Hello and welcome to our presentation. We are Group 9 and our chosen topic is Inside Genomes of a Dinosaur. In this video, we will explore how bioinformatics tools were used to uncover the hidden genetic informations preserved within these ancient remains. Our team has worked together to compile this analysis and we will guide you through each steps of the process, from genome sequencing to the identifications of closely related organisms. And our chosen article is Genome-Centric Resolutions of Novel Microbial Lineage in an Excavated Centrosaurus Dinosaur Fossil from the late Cretaceous of North America. But before we get started, let me introduce you to my group members. Hi, my name is Ahmad Shawal bin ADNAN. Next is Iqbal Hakim bin Jamaluddin, Ahmad Muhammad AL-AMIN bin MATERANG, Damia BALQIS, and lastly is Siti Nur Alia SHAMIRA binti Muhammad Noor. Without further ado, let's get started.
The study finds 46 high-quality metagenome assembled genomes (MAG) from different bacteria and archaeas group. These MAG belong to the bacteria phyla Actinobacteria, Proteobacteria, Nitrospira, Acidobacteria, Gymnomonads, and Chlorophyllaceae, as well as the archaea phylum Taumaceata. These phyla contain the species that are most closely linked to the identified microbial lineage. Notably, some MAG from the classes Nitrileuctoria, Delta Proteobacteria, and Beta Proteobacteria were more abundant in dinosaur-born samples than in the surrounding mastodon, indicating that these groups are especially close cousins. This shows that the microbial community in dinosaur-born differ from those in the surrounding environment, with specialized lineage switched to the bones, particular circumstances. The research article Genome-Centric Resolution of Novel Microbial Lineage in an excavated Centrosaurus dinosaur fossil born from the late Cretaceous of North America provide information on various metagenome assembled genome (MAGs). For example, assembled genome MAG. For example, the research article Genome-Centric Resolution of Novel Microbial Lineage in an excavated Centrosaurus dinosaur fossil born from the late Cretaceous of North America provide information on various metagenome assembled genome (MAG). For example, one of the detected MAG is accessible at NCBI with a genomic size of 2.5 MB and accession number GCA-0099222785. The genome of a different MAG, accession number GCA-0099227595, is 3.1 MB and may be found at the NCBI. Other MAG are available through the individual NCBI URS Biomed Center at small, one with a genomic size of 2.8 MB, GCA 0099222805, another with 3.4 MB, GCA 0099222815, and and a with 2.7 MB. GCA 0099228025. This is the figure show accession number in article is 46 MAG. And then from this link we found this article. This is the data availability statement and the accession number represent the total of 46 MAG.
Next, let us dive into the various bioinformatics tools used in these articles. There are 5 tools that is used for co-assembling and binding process, which is MegaHit, MetaBed, MaxBin, Concord, and MetaWrap. First, let us dive into MegaHit. Used for assembling the metagenomic data. MegaHit is an efficient assembler for large and complex metagenomics dataset. It handles high-throughput sequencing data to reconstruct the genomes of the microorganisms present in the sample. Next is MetaBed, MaxBin, and Concord. These 3 tools have same purpose, which is used for binding, which is the process of grouping assembled sequences into individual genomes. MetaBed function is used in metagenomic read, depth, and sequence composition to bind contigs, while MaxBins utilize an expectations maximizations algorithm to cluster contigs based on tetranucleotide frequencies and coverage, while Concord combines sequence compositions, coverage data, and linkage information to cluster contigs into bins. Lastly, for MetaWrap, its purpose is a comprehensive pipeline that integrates various bioinformatics tools for metagenomic data processing. MetaWrap provides modules for read quality control, assembly, binding, bin refinement, and more. It leverages multiple binding algorithms to improve the accuracy of genome recovery. Furthermore, for metabolic annotation and phylogenetic analysis, there are 2 tools that are used. First is CheckM and second is SPADES. The first one, CheckM, used for assessing the quality of metagenome assembled genomes max. Its function, CheckM evaluates the completeness and contamination level of the max by comparing them against a set of single copy marker genes. Finally, SPADES. SPADE purpose used for genome assembly. SPADE is another assembler used to handle various sequencing technologies. In this study, it may have been used to assemble specific genome regions or to refine assemblies. There are a total of 7 tools used in these articles.
So today I will discuss about the advantages and disadvantages tool used in this article. The first one is MegaHit. The advantages of MegaHit is ultra-fast assembly. MegaHit is designed to be fast with assembly time measured in seconds compared to minutes for other tools. Next is low memory requirement. It is memory efficient, making it suitable for systems with limited resources. And lastly, fewer misassemblies. It's time to produce fewer misassemblies compared to other tools like SPADES, especially at low coverage. But for the disadvantages, limited performance on the high coverage data. MegaHits may not perform as well on the high coverage genome, potentially leading to a disadvantage for very large dataset. Next is assembly quality. While MegaHit performs well, it is outperformed by tools like ADPA and SPADES in terms of assembly quality, particularly at the high coverage. Third is digital normalization issue. Using digital normalization with MegaHit can lead to misassemblies, especially on the high coverage genomes. And lastly, rough edges. The tool is still in development and may have some raw edges, requiring users to be familiar with command line interface and UNIX system. Next is MetaBuds MaxBin and Concord. The advantages of this tool is computational efficiency. This tool, known for its fast computing speed and low memory requirement, it can even be faster if multiple rounds of binning are performed to fine-tune parameters on the large dataset. Second is high accuracy. It outperforms other automated tools in accuracy, particularly in identifying unique genome bins that other tools miss. And last is scalability. It can handle a large dataset efficiently, including those with a million of content, and can recover hundreds of high-quality genome bins in a matter of hours on a single node. But for other disadvantages, parameter optimization. This tool does not automatically choose binning parameter based on the underlying data. User must select from the phrasal condition to achieve the best result. And lastly, dataset choice. The performance might vary depending on the specific microbial community being studied. It is advised to evaluate different binary on the specific dataset for performance comparison or to combine results from different tools for comprehensive binning. Next is MATERANG. The advantage is flexibility. MATERANG allows users to control the analysis process while being easy to install and use. It includes hybrid algorithms that leverage the strength of various software to extract and refine high-quality bin from metagenomics data through bin consolidation and assembly. Next is improved bin quality. The bin refinement module uses a novel hybrid approach to consolidate bin predictions from different binning software, producing a single stronger set. This approach significantly outperforms individual binning software and other consolidation algorithms, especially at the highest completions and lower contamination range. Lastly is modularity. Each module of MATERANG is a standalone component, making it a flexible and versatile tool for tackling metagenomic shotgun sequencing data. This modularity allows users to follow intuitive workflow or use specific function as needed. The next is disadvantages of the complexity. MATERANG involves a collection of modules, is addressing a specific aspect of the BioMG data processing or analysis. This complexity may make it challenging for users without an intensive bioinformatics experience. Second is time-consuming. The pipeline can be computationally intensive, particularly for the large dataset. The majority of the compute time is dedicated to the recursive binning, bin refinement, and resemblance bin module. Lastly, is dependent on specific software. MATERANG relies on the various installed software and custom Python script, which may require additional setup and configurations. Next is checksum. The advantages of the checksum is accurate estimates. Checksum provides robust estimate for the genome quality, which is essential for making biological inferences from draft genome. Second is lineage-specific marker genes. The tool uses marker genes specific to a genome, inferred lineage within a reference genome tree, which provides a more accurate estimate compared to universal or domain-level marker genes. And lastly, improved quality control. Checksum can identify problematic genomes before they are deposited in public database, ensuring the quality of genome used in research. But for the disadvantages, limited applicability. Checksum primarily designed for bacterial and archaeal genome and may not be suitable for the aquatic or fresh genome. And dependence on reference genomes. The tool relies on the comprehensive reference genome tree, which may not be available for all lineage, limiting its effectiveness in certain cases. And lastly is a SPADES. The advantages of this tool is speed and accuracy. SPADES is known for its fast and accurate assembly capability, making it a popular choice for bioinformatics application. Second is customizable. It has a simple command line interface that accepts various options and parameters to customize the assembly process. And lastly is multi-threading support. Spiders support multiple threads, allowing it to utilize available memory and CPU resources efficiently. For the disadvantages, memory requirement. Spiders require a large amount of memory to perform assembly, which can be challenging for users with limited computational resources. And lastly, no hybrid assembly. Spiders does not support hybrid assembly, which means it cannot combine RAID from different sequencing technology to improve the assembly quality and accuracy.
Analytical method. First, sampling site and fossil bone excavations. In 2016, at Dinosaur Provincial Park, Alberta, Canada, a Cetrosaurus apatus fossil from bone bed BB118 was successfully excavated and transported to Princeton University for DNA extractions. Sample was stored at -18 degrees Celsius to ensure preservation and prevent contamination. Secondly, bone pre-treatment and geochemical characterizations. The fossil bone and surrounding mudstone underwent meticulous separations, scraping, and powdering before being analyzed for water extractable anion using ion chromatography. Number 3, DNA extractions and metagenomic sequencing. DNA was extracted from bone powder, EDTA, treated bone powder, and mudstone using the DNEZ PowerMax soil kit. The extracted DNA was prepared into libraries with unique barcodes and sequenced on Illumina HiSec 2500, generating three metagenomics with around 120 million pair and reads in total. Next, for number 4, contents, assembly and reconstructions of MAG. Raw sequences were filtered and assembled into contents using megahit, which were then grouped into MAGs using MetaBed, MaxBin, and Concord. In MetaRep, MAG, meeting quality standard, were refined and reassembled with spades, finally assessed with check-M for high quality MAG selection. Next, number 5, metabolic annotations and phylogenetic analysis. Genes in MAG were annotated using PROCA and devised against TIGR, FAM, and COG databases. Enzymes were confirmed via BLASP. A metabolic pathway for carbon and nitrogen were predicted using RAS and KEGG plus QUALA. Phylogenetic analysis used 16S ribosome RNA genes identified by ENVIO and RNA-mer concatenated ribosomal protein were aligned with muscle trim with TRIM-AI and analyzed with RAXML and iTool for visualization. Number 6, other genome-centric analysis, taxonomic classification of MAG using GTDP, TK, novelty assessment via average amino acid identity, AAI, quantification of relative abundance with MetaRep, identification of unique functional gene cluster using MVO, and estimation of microbial growth rate using growth rate index GRID based on coverage ratio at replication origins and terminus regions, validated by DNA and different sequence. Reanalysis of 16S ribosome RNA gene amplicon data. The study reanalyzed 16S ribosome RNA gene amplicon data to compare microbial abundance between dinosaur bone and adjacent mudstone sample using statistical tests. A canonical correspondence analysis, or CCA, was employed to explore correlation between microbial community composition at the class level and geochemical parameters using R software with Bray-Curtis, distance and permutation tests for ordinations. Lastly, metaproteomic analysis. Various methods were used to extract proteins from fossil bones and adjacent mudstone due to low biomass challenge. So this method include chemical dissolutions, SDS buffer, extraction, and a commercial kit. Identify proteins underwent mass spectrometry analysis for identification against database. So this is to confirming their presence with at least one unique peptide per protein. This is analytical method in a flow chart. So first, sampling site and fossil bone excavation. Secondly, bone pretreatment and geochemical characterization. Next, DNA extraction and metagenomic sequencing. Fourth, contents assembly and reconstruction of MAG. Next, metabolic annotation and phylogenetic analysis. Then ada genome-centric analysis. Number 7, reanalysis of 16th as ribosome RNA gene applicant data. Lastly, metaproteomic analysis.
This figure shows how different microorganisms are related based on 16 ribosomal proteins. Black star mark ocean microorganism, while red star mark those from very salty place like Salt Lake. Purple dot means strong branch support above 70%, while branches without dot have weaker support below 70%. Names starting with P are phyla, and those with C are classes. The scale bar show 0.1 change per amino acid position, indicating genetic differences. This figure displays the relative abundance of MAG in three metagenomes: one B5G, one beta, and one M10G. The abundance is calculated based on how many reads map to each MAG and is normalized to the sample size, showing genome copy per million read. This help compare the efficiency of different microorganisms across the three samples. This figure shows a phylogenetic tree created from 16 sRNA genes of MAG and their close relatives, including both culture and uncultured organisms. Black dot indicate not with bootstrap value greater than 70%, which were determined from 1000 replication. The 16 sRNA gene from MAG and this study are highlighted in bold and red. The scale bar represents 0.01 substitution per nucleotide position. This figure shows the presence of various functional genes indicated in red in MAG that help them cope with high osmolality in a dinosaur fossil bone. Figure 5 illustrates potential biogeochemical pathway mediated by various MAG found in the Centrosaurus dinosaur fossil bone. MAG marked in red are involved in autotrophic CO2 fixation pathway. These lines show where key genes for this pathway were not found in the MAG.